So today we are going over the process of mitosis, and I'm going to be explaining that process with a visual representation here. So right here I have this white circle, which is going to represent the entire cell, and these three Legos are going to represent the chromosomes that are inside of the nucleus. So there's two parts to the process of mitosis. And that is what's called the interphase and the mitosis phase. And then these two phases are broken up into multiple phases as well. So we'll start with the interphase. So the first part of the interphase is what's called the G1 phase, and this is where the cell, so when a new cell is born, it'll start to grow and get bigger. I don't really have any way to represent that. But that's what happens. And then the next part of the interphase is what's called the synthesis phase. And this is where DNA becomes duplicated. And so right now the DNA is packaged in these chromosomes in the nucleus. And so what happens is these chromosomes are going to each replicate themselves. And so to represent that, I just have these other Legos that I'm going to attach on each one. So these chromosomes are just duplicating themselves, but they're still attached. And so each of these chromosomes is going to have two parts, and they're called sister chromatids. So that's the synthesis phase. And then in the G2 phase, which is the next part of the interphase, this is where the cell continues to get bigger and final preparations are made for the cell division. And some of those other preparations include the separation of what's called centrosomes, which are organelles found near the nucleus, and these organelles will separate and move to opposite sides of the nucleus. So that is interphase. The next part is the mitosis phase. And like I said, mitosis phase is also broken up into different phases or different parts. So the first part of the mitosis phase is what's called the prophase. And in the prophase, the chromosomes become thicker and shorter, and to the point where you can actually see them under a microscope. And I think I already mentioned that the centrosomes move to the opposite sides of the nucleus. And then that's the prophase. Next part is the prometaphase, and this is where the nuclear membrane actually disappears, and that will give those two centrosomes access to those chromosomes. And then these proteins called spindle microtubules now have access to those chromosomes. And so I don't really have a way to represent that, but basically what's going to happen is each of these chromosomes is going to form a protein structure called I always pronounce this one wrong too, kinetochores. I believe that's how you say it. And so those spindle fibers are going to attach to those kinetochores, and that's what's going to pull these chromosomes apart. And that brings us to the next phase, which is metaphase, which is where these chromosomes will line up. I kind of already had it set up now that I'm realizing, but these chromosomes would have been before just kind of like in a mess like this. And in the metaphase, that's when the chromosomes are going to line up and align like this with the help of those spindle fibers. And then comes the anaphase, which is where these chromosomes break at the centromeres. So I'll just break each of these apart. So the sister chromatids of each chromosome break apart, and those spindle fibers will pull the sister chromatids to the opposite sides of the cell as the cell starts to divide. So they're going to be pulled to the opposite sides like that. And then the next phase is the telophase. This is where the chromosomes will uncoil and the spindle fibers disintegrate, and two new nucleuses will form. So as the cell is splitting, these these chromosomes will just uncoil, I guess is the best word. So they'll just kind of become a mess of chromatin again. And two new nucleuses will form as the cell is splitting. And then the very last phase is what's called cytokinesis. And this is where the cell is like finally broken apart. And it's done by, let me see, I always forget. Okay, so myosin 2 and actin filament ring contracts right here in the middle of the cells, and then that ring kind of just like closes and it squeezes the two cells apart. And at that point, we have two cells with exactly the same DNA, and mitosis is complete.